So welcome everyone. Thank you again for coming to this edition of the seminar series. So Dr. Lacal is a researcher in genetics and molecular biology. He has a PhD in biochemistry and molecular biology from the University of Granada. During his thesis, he did the biochemical and molecular characterization of the two-component system TOD-S and TOD-T of Pseudomonas butida, which is a system that regulates the expression of genes involved in the degradation of hydrocarbons like toluene and benzene. After that, he went a few years in the United States, especially in the group of Dr. Richard Fertel in the Department of Cell Biology and Development of the University of California, in which he worked on the chemotaxis involved in cell polarization and cytoskeletal reorganization. Then he came back to Spain as his current position, Associate Professor of Microbiology and Genetics at the University of Salamanca. And with his research group, Functional Genetics of Rare Disease, he works, on rare disease and rhizopathies in particular, which are a group of syndromes caused by changes in genes involved in the Ras-MAP kinase pathway. So I will now leave the floor to Dr. Lacal for his talk called Reproducing Patient-Specific DNA Variants for Cell and Molecular In Vitro Analysis. Thank you, everyone.
Thank you so much, Madeleine and Marco, and thank you everyone who made possible to bring me here to Paris this week. I'm still super happy and very honored to have the opportunity to show you what we are doing in my lab. So the title of the talk is Reproducing Patient-Specific DNA Variants for cell and molecular in vitro analysis. So already Marco mentioned in the introduction, I'm a professor in genetics in the Division of Genetics at the Faculty of Biology, University of Salamanca. I'm also the exchange departmental coordinator of the Faculty of Biology. So I wanted to mention that just in case any students are interested in starting to collaborate within the bilateral Erasmus agreements that we have with the University Paris-Cité starting this year, which is actually thanks to that new agreement, I'm here. So I welcome everyone who is interested in either studying or doing a traineeship abroad to consider the Faculty of Biology at University of Salamanca. So feel free to contact me for further questions or Professor Domenico, all right, from your university. That being said, I would like to first start introducing my group. It's an emerging group. We started 3 years ago in the University of Salamanca. This is me on the very left, and so far I have 3 PhD students, starting with Juan Baez, Mario Rodriguez, and also Noemi Ferrito, who is the last PhD student that joined my lab last September. I also have this year 2 master students, Alessio Coppola and Christian Codel. And I also have a bunch of undergraduate students, including 3 undergrads who are carrying out their practical dissertations in my lab. In the picture is one of them, Julian. And I normally or usually also have some international students, usually from Germany, such as Leonie. They come with a DAAD program, DAAD program, which usually funds like outstanding German students to study abroad. And also I have students from the Erasmus program. Normally they come either from France or Italy so far. Although next week, one Portuguese student will also start in my lab, also with the Erasmus program, first time a Portuguese in the lab. So that means I like to keep the lab international, so students practice their English, and they also get to meet other peers from other countries, mostly European countries, and I think that's a really good benefit for everyone. Apart from people I mentioned already, we also have 2 lab technicians, Sonia and Fernando. And then, importantly, we have the collaborators. The Spanish collaborators include Maria Isidoro and Pablo Prieto. They both are medical doctors from the University Hospital of Salamanca. And as I will mention later, thanks to them, we can directly study DNA variants that are found in pediatric patients, specifically pediatric patients. I also count with Raquel Spin. She is a professor in Iowa State University. It's a recent collaboration, as well as the collaboration with Vanessa Rives from the Institut Jacques Monod, who I think is present, is in the house. And I will mention later what these two collaborations have to do with the rhizopathies and with my lab. Just a quick view of the campus of the University of Salamanca. It has different campuses. One of them is the biomedical campus, which is called the Campus Unamuno. The Campus de Unamuno is composed by 3 buildings: the Faculty of Biology, which contains 3 different buildings, the Cancer Research Center, the Faculty of Pharmacy, and also the Faculty of Medicine, among others. And then there are 2 huge hospitals. One of them is the University Hospital, which allows a close connection and collaboration with medical doctors and patients. We also have the downtown of Salamanca, and there are other buildings in the spread across the city, although they are not related to the biomedical field. This is the departmental building, which is the actual building where my lab is located at, in the 3rd floor. It's a really nice building, and in my lab we have room up to 10 people. In today's talk, I'm going to talk about rare diseases, focusing on rhizopathies, and then we'll go through what are the major research questions to answer in the field and that we are trying to study and push further. I'll show some of the experiments we're carrying out so far, and then I will mention what are our future plans. Starting with the rare diseases, a rare disease in Europe is considered a disease that affects fewer than 1 in 2,000 people. Although rare, in total, in Europe, they are estimated to affect around 30 million people, 300 million people worldwide. There is no cure for the rhizopathies for most of them, and there are only a few treatments available. And treatments usually target symptoms of the disease, but they don't really target the root of the disease. Most of them, about 70 to 80% of the rare diseases, are genetic. They do have a genetic cause, and they also the onset of the disease is in childhood. As a recommendation, if anyone is interested on rare diseases or want to look for more information, I strongly suggest to look at the online portal for rare diseases called Orphanet. In Orphanet, this is updated. We have more than 6,000 different rare diseases annotated, corresponding to nearly 6,000 different genes. And then you have numbers that correspond to research centers or universities working on rare diseases, diagnostic tests, professionals involved, and also like a count daily visitors icon at the very end. As their website reads, rare diseases are rare, but rare diseases patients are numerous. And that's why there are several organizations who are supporting research towards finding cures for rare diseases, including the World Health Organization, the International Rare Diseases Research Consortium, as well as EUR G.E.N.E, rare diseases in Europe. At the national level, we count with 2 major organizations. In France, you have the Alliance Maladies Rares, and in Spain, we have the Fundación FEDER. It's like FEDER Foundation for the Investigation of Rare Diseases. They both share the same concept. They are basically formed by patients and patients associations who are trying to support with funding and with publicity the study and the research of rare diseases found in these countries. So also feel free to go to their websites and have a look at what they're doing to incentivize research on rare diseases. In my lab, we're particularly interested in the RASopathies, and this is a scheme of all different RASopathies, most of them that have been studied so far. The RASopathies are neurodevelopmental syndromes that arise from genes or alterations in genes that encode a component of the RAS-MAP pathway. So they share, or they have in common, that they affect the RAS-MAP pathway. Therefore, the name of RASopathies, because the major protein that controls the RAS-MAP pathway is the small GTPase called RAS, RAS. Within the RASopathies, we have a bunch of different syndromes, including, or I'm going to highlight, neurofibromatosis type 1, which is caused by the NF1 gene, mutations in the NF1 gene. So this is a monogenetic disorder. Then we have the Noonan syndrome, which can be caused by multiple alterations in genes, including or mostly PTPN11. Almost 50% of Noonan syndrome patients have mutations in PTPN11. And then Jesús syndrome, which is considered an ultra-rare disease and is also a monogenetic syndrome caused by mutations in the gene SPRED1. As you can see, there are a few other RASopathies, but so far we are focusing on those three. The reason we are studying those three is because based on the pediatric patients in the region of Castilla y León in Spain, most of the patients have been diagnosed with either neurofibromatosis type 1 or Noonan syndrome. Up to 80% of our patients fall into one or the other category of these RASopathies. And we have some patients also with Jesús syndrome. And that was interesting because there are not many papers out there reporting study cases on mutations in Jesús syndrome patients. Most of these syndromes have autosomal dominant inheritance. That means that just one copy, one altered copy is enough to cause the disease. Although two autosomal recessive genes have been also found, LCTR1, which cause Noonan syndrome, and SPRED2, which cause a Noonan syndrome-like. Just a quick reminder, as I was told that many students are probably watching this, just wanted to briefly remember that the autosomal dominant means that just one copy is enough to cause the disease. And that's usually one of the characteristics that the pediatricians take into account when they have a kid with a suspicious RASopathy. They also study the parents and see whether one of the parents, either the mom or the dad, contains a mutated gene, because that's also one of the features or characteristics that are taken into account for the diagnosis of a RASopathy. The RASopathies, as I mentioned earlier, they share in common that they affect the RAS signaling pathway. Here on top of the slide, you have the cell membrane. We have some receptors, mainly the RTKs, the receptor tyrosine kinases. These receptors get activated by growth factors, and then they will activate SHP2, which is the protein encoded by the PTPN11 gene that cause the Noonan syndrome. And then we have a bunch of other genes that will end up activating RAS. RAS is a switch in the cells. It's normally in the GDP state, but it can get activated by switching the GDP molecule for GDP, becoming activated. So think of RAS as a switch. This is the off state when it's GDP bound, and then it's on state when it's GDP bound. This particularly exchange is taken care by GEF factors, guanine nucleotide exchange factors, including SOS is one of the proteins that facilitates the exchange of GDP for GDP. On the other hand, you have proteins that are going to inactivate RAS. Those proteins are called GAPs, GTPase activating proteins, because what they do is to accelerate the intrinsic GTPase activity of RAS, and therefore hydrolyzing GTP for GDP, becoming inactivated. One of the GEF proteins is NF1, neurofibromin, which is one of the genes we're also studying. And then we have the other gene we're studying, that is spread, that encodes the spread1 protein. And this protein interacts with neurofibromin and controls its localization to the cell membrane. Because most of these proteins, particularly when they are activated, they work at the cell cortex, cell membrane, near to the inner cell membrane. When RAS is activated, this protein is going to signal downstream proteins by phosphorylation events. And starting with RAF gets activated and will phosphorylate MEK, and then MEK will phosphorylate ERG. Apart from the classical RAS-MAC pathway, which is this one, RAS is also able to control another major signaling pathway that it's called AKT-mTOR pathway. Both pathways are responsible for mainly cell proliferation, cell survival, growth, and cell migration. Therefore, any aberrant activation of these pathways usually triggers important cellular defects, including excessive cell proliferation or overgrowth of the cells. That would be the case if NF1 is mutated. If NF1 is mutated, as an example, RAS will be way more activated, and therefore the phosphorylation of downstream proteins in those signaling pathways is supposed to be much higher. And the cells will have aberrant RAF, MEK-ERG, and AKT-mTOR P70S6K activation. The epidemiology of the RASopathies is known for Noonan syndrome and neurofibromatosis type 1, and those two are rare diseases. Their incidence is between 1 in 1,000, 1 3,000 people. And then for the rest of the RASopathies, the epidemiology is unknown because they are too rare and there is no published data regarding those RASopathies. In terms of the association with ethnic origin, there is no known association so far. That means that it affects, in principle, any kind of people depending on, regardless of the ethnic origin. And also men and women are affected equally. There is no difference. Regarding the RASopathies, because they share the same signaling pathway somehow, they have they are characterized by having overlapping features. One of the major overlapping features is the short height that is present in these people affected with RASopathies, dysmorphic facial characteristics, the congenital heart disease, the lymphatic dysfunction, and also learning or intellectual disabilities. Apart from these overlapping features, there are also specific clinical features affecting patients with RASopathies, including defects on the central and peripheral nervous system, the endocrine system, gastrointestinal, skin, virtual system, cardiovascular, and musculoskeletal system. So as you can see, the RASopathies can be multisystemic disorders, and then dependent on the specific variant or of a specific gene, the symptomatology of the patients will be different. As I said in the beginning, the RASopathies are rare diseases, have no cure, although there are some treatments available, including growth hormone therapy for those people who present a short height, surgery, particularly in the case of those who present solid tumors. Then we have medications, as I said, to usually palliate the symptoms of the RASopathies, such as pain, headaches, seizures, or so on. There are also, it's important to have a physical therapy, particularly to improve the musculoskeletal ability and the mobility of these people. And last but not least, it's important to monitor and surveil the patients, because through development and through the years, the symptoms will likely vary. So then the doctor needs to revise the treatments, the medications, or evaluate whether or not surgery is needed. So with this introduction, I want to get to the point to what is the scientific community trying to study these days, and what are the major goals in the RASopathies. And basically, now that thanks to the next generation sequencing techniques, it's quite likely to identify the cause of the disease. Now, the research community is moving towards understanding the genetic, the genotype, phenotype correlations of these diseases. So once we have a gene mutated, people want to understand what would trigger in the patient. So in order to do that, we need to do basic research to understand what is the functional effect of the different DNA mutations that are found in these patients. What's the goal of all this? To treat or delay the progression of the rasopathies at the earliest point possible. That's why in my lab, we have started to work with these three genes, NF1, SPRED1, and TTPN11, in order to first understand the cellular mechanisms behind these DNA variants identified in pediatric patients. Then we will continue by understanding the molecular mechanisms behind those changes in the cells. And last, this is more like a mid-long-term goal for us, will be to validate and try FDA-approved drugs, both in vitro and in vivo, to see if we can offer a more personalized precision medicine to the patients based on the genetic diagnostic. How we work in the lab, basically, how the whole story starts is that when families with kids that probably have rasopathy or rare diseases, they go to the hospital. They are seen by Pablo Prieto, which is the head of the pediatric unit, and then they will evaluate the kid. If they suspect the kid may have a rare disease, then one of the first things they will do with this patient is to take a blood sample, and then they will analyze the exome, the whole exome. So therefore, they carry out whole exome sequencing, and they can analyze about the 20,000 different genes that we have in the human genome. If they do not find any changes in the DNA that could be suspicious of a rasopathy, they can also perform whole genome sequencing to also sequence like introns and other regions that do not encode proteins. Once they have the genetic information, they carry out a complex bioinformatic analysis to eventually suggest one or several potential variants that are the cause of the disease. But at this point, sometimes they are not completely sure whether or not the variant is the cause of the disease. That's why it's important to go to the literature, to the scientific literature, and also to carry out basic research studies in a lab to identify whether or not that variant is pathogenic. And that's where my lab enters basically in this idea of studying the rasopathies. We get the DNA data from the hospital. From the DNA data, we carry out some in silico analysis that I will explain further later. Then we carry out genetic engineering analysis to reproduce those variants in expression lentiviral vectors. Then we'll carry out cellular biology to generate stable cell lines expressing those variants. And then we will carry out molecular biology analysis to study the signaling pathways that are potentially affected, such as the RAS-MAP pathway. The idea is to have a full array of data, and then by combining the data from the patients, the genetic analysis, and all the molecular biology and cellular analysis to be able to offer this precision medicine to the patients. This is the kind of information that we start working with in the lab. So this is an example of one of the tables we can get from the pediatrician unit. It's basically, as you see from, you can read from left to right. We have the gene information. We have the number of the exon that has been affected, the type of variant that has been identified with the next generation sequencing and confirmed by Sanger sequencing. Then the specific information of the nucleotide that has changed and to what has changed, and whether or not it's a novel mutation or if it has been inherited from the mother or the father. And then last, we usually include data from different databases that predicts the potential pathogenicity of the variant. This information, along with the clinical information, will be put together to try to understand if there is a correlation between the genotype and the phenotype of the patients. The in silico studies, as I mentioned, normally we use 3 different databases or platforms. Varson is one of them. Varson is basically a database. It's free for everyone. You can go online, enter Varson in Google, and you can log in, preferably, and then you have access to the study of what is the potential impact of any variant in the humans in terms of a phenotypic outcome. This program runs several, let's say, like databases or other programs that kind of predict whether or not a variant is pathogenic. And probably the most important one, the one I will recommend to focus, is this one in the center called ClinVar. Okay, so Varson also gets data from ClinVar, which is another huge database that basically has information on the prevalence of a certain DNA variant, human variants. And as you can see, for instance, ClinVar predict that this particular variant in NF1 gene is likely pathogenic. Then another database that we use is Cosmic. This one is particular to study somatic mutations found in human tumors, and therefore is to study cancer. The rhasopathies are rare diseases, and of course they have their symptoms, but they also, most of them, has an increased predisposition to tumorigenesis and to suffer from cancer. So therefore, we're interested in both studying the symptoms of the rhasopathies as rare diseases, but also why sometimes some of these variants can trigger cancer in these patients. So the idea is kind of the same as Varson, to enter the specific mutation you are studying and see whether or not this mutation has been previously identified in human tumors. And then whether or not this particular mutation has been identified in human tumor, we still use this database to collect information regarding the different type of mutations and where have been found, not just in terms of the gene, but also in terms of the tissue and the histology. And the last program I wanted to mention in this talk is particularly very useful for students. It's called Serial Cloner. Serial Cloner is a free software. That means you can download it from the internet, and it works in any platform, from PC to Mac to Linux. And it's basically a program that allows you to edit and work with DNA and protein sequences, but particularly with DNA sequences. And you can do from recombination analysis to PCR, in silico, of course, to genetic engineering. You can do a lot of cloning related things, alignments with DNA, and so on. So very useful for those who are studying genetics and want to include an extra support to their studies. And also very useful for the lab. Like all the people I have in the lab use Serial Cloner to design their genetic engineering strategies. So I strongly recommend to look it up and download it. And feel free to ask me questions for Serial Cloners either today or further in the future if you are not sure how to use it. But it's very user-friendly, so it's really easy to use. Here I'm presenting some data, general data, just to give an idea of the type of variants we're working with. In the case of NF1, we are studying, as you can see, like 5 variants were previously reported, whereas the 4 variants we have not been previously reported. So these are new variants that we have identified in neurofibromatosis type 1 patients. Then for the PTPN11 gene, most of the mutations we have been previously reported in the literature. We have 9 variants, and one of them is new. And for a spread, we have 5 variants previously reported and 6 variants that are new. So in total, we have 30 different DNA variants that are under study in my lab. Regarding the impact or whether or not they have been previously found in tumors, 3 out of the 9 samples or the 9 variants in NF1, 3 of them have been found in tumors, 4 for PTPN11, and 4 also for spread1. So as you can see, usually some of the variants seems to have a higher impact in tumorigenesis than other variants. Also, some general data, or this is one of the analysis that one could do by using our downloaded cosmic data, is that in the case of, this is particularly for neurofibromatosis type 1, for the NF1 gene, all the acronyms you have here correspond to different domains in neurofibromin. And then what we are doing is to basically localize the type of mutation in the different domains and see whether there is a difference in the distribution or the different type of mutations, such as missense, nonsense, insertion, deletions, or coding in across the NF1 gene. As you can see, there is no obviously particularly distribution of one type of mutation in a different domain, meaning that probably any missense or nonsense mutation could be found across the protein in particularly in mutations found in tumors in patients. And this is the top 3 of tumors that have been found. So we have, this is a simplified table that we've been working on, basically analyzing all different, more than 5,000 different variants in NF1 found in tumor patients. And the first column correspond to the 3 most frequent type of tissues affected, primary tissues, which are skin, lung, and breast. The subtype tissue more affected or with more mutations in NF1 gene is the brain, followed by nerve sheath and bladder. Regarding the histology, the carcinoma is the most abundant type, followed by malignant melanoma and glioma. And considering the histology subtype, the most abundant ones are adenocarcinomas, squamous cell carcinoma, and astrocytoma grade 4. So these data are based on what type of mutations and the number of mutations that have been found in those tumors. And these are the most abundant ones. It doesn't necessarily mean that NF1 is the cause of these tumors, but at least it was found mutated in those tumors. So it's a way to start making correlations between the genotype and the phenotype, although of course, all the genes could be also mutated. Regarding the genetic engineering part, I'm going to present an example of what is our workflow in the laboratory. This is the example of the cloning of the PTPN11 wild-type gene. We normally start with commercial plasmid that we get from databases such as @Gene to get the wild-type copy of the gene, usually without any introns, or just the cDNA. And then we carry out a PCR to basically add upstream and downstream the gene, the restriction enzymes that we need in order to do the final cloning into the lentiviral expression vector. But first, what we do is the PCR product is directly cloned into a linearized cloning vector. We normally use the PBSK vector. This one is linearized, and the PCR product is cloned directly into that vector. We sequence the DNA to make sure no extra or non-wanted mutations are there. And then when we verify the sequence is the right one, we clone the wild-type gene into the expression, the lentiviral expression vector, which is like the final construct that we need. Also, once we have the wild-type copy in the cloning vector, by site-directed mutagenesis PCR, we basically regenerate the DNA variants that have been identified in the pediatric patients. So that's why we can reproduce those variants in these plasmids. And once again, from the cloning vector, we can cut and paste the gene, clone it into the lentiviral expression vector. The lentiviral expression vectors that we have generated so far are in total 33. 10 for the NF1 gene, 11 for the PTPN11 gene, 12 for spread1. We have also generated as a positive control for tumorigenesis and an expression vector that expresses the oncoversion of HRAS G12V, which is a good oncogene to use as a control in your experiments, cell culture experiments mostly. The lentiviral vector is an expression vector, and we work with the second generation lentiviruses, which means that we use another two vectors that contains all different genes that would allow the virus to be produced inside the cells. So basically, we combine these three plasmids, we transfect them into 2933 cells, and then we let the virus to generate. Once the virus are generated, we're going to collect them, and they will be used to transduce different type of cell lines, including 2933 cells and others. This is an example, a brief summary of the transduction and the infection with the lentiviruses. So once we have the lentiviruses, we're going to use 6 well plates in a condition. We won't add the lentiviruses, so that will be used as the control that the cells basically should die after the treatment with the hygromycin, the antibiotic that we use for the selection of the cells. I can explain more after the seminar if you're interested on how this works, the selection, but for now, I will just skip that part. But basically, in each well, we apply a different treatment, either no virus or a virus that contains the empty vector, that we call it MOC, the vector expressing the wild-type protein, or the vector expressing the different variants. After 24-hour incubation with the viruses, we continue with the selection with hygromycin, which is a treatment that takes between 7 and 14 days. After that treatment, the cells that survive are cells that have integrated the viral DNA in their genome, and therefore also express your gene of interest. Then the cells are freezed and kept in liquid nitrogen until further use for cell or molecular biology analysis. So far, we've been able to generate all different stable cell lines for NF1, PTPN11, and for spread, and also the new oncogenic gene that we recently generated in both the HEC293T cells and also in the NAH333 cells. We use so far these two different cell lines because the HEC has been proposed as a good model to study RASopathies, and historically is one of the cell lines that have been used the most to study RASopathies. And then on the other hand, we also have the NAH333 cells, which is a murine model that is very good to test tumorigenesis and cells transformation to study oncogenesis. This is a scheme we're interested in. Neurofibromin, which is a huge protein, is one of the largest protein in the cells. It has nearly 3,000 amino acids. As you can see, it contains different domains. We have SHP2, which is a phosphatase. Its major role is to dephosphorylate tyrosines and activate RAS. Neurofibromin, as a quick reminder, basically inactivate RAS. And then spread, that contains 3 different domains. It binds neurofibromin through the N-terminal domain, and it can then translocate neurofibromin to the membrane thanks to the SPR domain. This is a figure we have generated in a paper that is right now under review in communications biology, and hopefully we expect it to be dissected soon. And it basically recopilates all different signaling pathways where neurofibromin has been involved. As you can see, neurofibromin, it can translocate to the nucleus, and it can regulate gene expression. And also during the cell cycle division, it regulates the localization of the chromosomes in the right position before the separation of the chromatids. Neurofibromin, it's also involved in degradation by the proteasome, and that's important for how the protein it's regulated within the cells. It's involved also in the pKa pathway, responding to cyclic AMP. It's also responsible for interacting with proteins that regulate the cytoskeleton of the cells, and the RAS-MAP pathway, the PI3K pathway, and other pathways such as ROCK involved in migration. So this is just like a quick summary of all the different signaling pathways neurofibromin can interact with. And as I said, we're particularly interested in studying the RAS-MAP and also the AKT pathway. And probably in the future, we will expand our studies to the remaining pathways. How do we study, or how do we continue in the lab once we have the stable cell lines? Well, one of the things we do is that we basically grow the cells under experimental conditions with low BSA, and then we extract the total protein from those cells and carry out SDS-based gels and electrophoresis. Then we transfer the proteins to membranes, and then we do Western blotting techniques with different antibodies to detect protein concentration and changes in protein phosphorylation. This is just an example of the type of data that we generate. We normally run as control the parental cell lines, so cells that have not been infected with the viruses. We also use as a control the cells that have been infected with the viruses to see whether that process had any impact on the generation of the stable cell line. Then we have the overexpression of the wild-type proteins, and then the overexpression of the different variants. One of the first plots that we carry out is, in this case, the detection of SHP2 protein encoded by the PTPN11 gene. So we want to make sure that we can overexpress both the wild type and all different variants. Probably depending on the variant, you may have more or less expression of the protein. This is just one plot, but we do at least these experiments in triplicate, and then we basically get the deviations and what's the average concentration for each particular variant to see whether or not, that's also that we should need to take into account how a particular DNA mutation can affect the concentration of the protein within the cells, because, for instance, could be more targeted by the proteasome. Once we assure that the cells were generated correctly and they are expressing the variants, we can continue, as most of you are probably familiar, by testing different antibodies such as phospho-ERK and, of course, the total amount of ERK. Apart from ERK, we also study MERK and AKT and MEK so far. The interesting thing is to theoretically see that the ERK protein levels, usually they should not vary from sample to sample, meaning that total ERK levels are not affected basically by the mutation. But sometimes, interestingly, depending on the variant and also on the conditions that you have, without inhibitors or without growth factors, you name it, you can see how the phosphorylation of these proteins is affected in the variants with respect to the wild type and with respect to the controls. And last but not least, we also include protein loading controls such as actin or beta-actin to make sure that we're loading the same amount of total protein for each cell line. The molecular studies are followed up by cell biology studies, including MTT assays, in which by a colorimetric assay, we can carry out like cell growth curves by measuring cell proliferation. The darker the color is, the more number of cells you have on your wells and then on your experimental conditions. So that way you can assess whether a cell line is growing faster or is growing slower than the controls when you are overexpressing a mutant protein. We also carry out cell morphology analysis to see what's the impact in terms of morphology, the size, the perimetry of the cells, and we can probably even in the future, the short midterm study some chemotaxis. That's kind of what I major in San Diego, in California, as a postdoc to study cell migration. We can also carry out, and we do carry out, what it's called the foci formation assays. This is a classical assay to test what's the impact of the DNA variant in cell transformation. As you can see in these two plates, you see like a clear background. That's because the monolayer of cells, basically the cells by cell-cell contact stop growing. They inhibit the cells to continue growing, and you have just one monolayer of cells that are stained, and then you see this blue color very homogeneous through the whole plate normally. But then if you are working with, if you look at down here, these are cells that are expressing oncogene, and when you have an oncogene, one of the characteristics of these cells is that they lose the cell-to-cell contact inhibition, and then they will continue growing, and then they will grow on top of each other. So the more cells you have, the darker and bigger the spots will be. By counting the number of spots and the size, then you can classify them in like non-tumorigenic or potential pathomorigenic, low, weak, mild, or strong tumorigenic potential, okay? Also including both negative and positive controls, such as oncogenic version of HRAS. And last but also not least, we can do the wound healing assays in vitro by growing the monolayer of cells, then make a scratch, and then see how the cells recover and they get back to filling the gap that was formed by the scratch. It's a way to study cell migration in these cells. I also wanted to mention that in the lab, we also can generate organoids. The type of organoids we're working with are epidermal for the human. And this is basically because
the epithelial cells of the epidermis undergo keratinization. The cells at the basal layer, the layer which is attached to the basement membrane and the connective tissue beneath, are cuboidal, and they divide frequently, maybe once a day. Once a cell is pushed up from this basal layer, it only has about 2 weeks to live. During this time, it synthesizes a great deal of the protein keratin, which is the major protein in skin, hair, and nails. Once the cell begins to push upward through additional layers, the cell gradually dies. The nuclei and other organelles degenerate, and so the cell then becomes a dead bag of the protein keratin. The cells are firmly anchored to each other because of the junctions found between these epithelial cells. These cells will typically stay on the epidermis for an additional 2 weeks, and therefore the outermost layer of our skin is made of dead interlocked cells known as keratinocytes.
All right, and we can generate in the lab those epidermis, as you can see on the top right, by staining. These are hematological cuts with the membranes that we can generate in the lab. On the left, you have a wild-type epidermis expressing a MOC, basically an empty vector. And then on the right, you have cells that have been infected with these lentiviruses and overexpress an oncogene. And you can see at the same magnitude how the cells are bigger. They have some of them, they have also different morphology. Most importantly, you can see how the number of layers of the epidermis also, it's higher. And sometimes also you see an impact on the keratin layers that sometimes is even thicker, or basically like thinner. Like it depends from variant to variant. So this is a really nice model to study. Like in our case, as you might remember at the very beginning, I mentioned that NF1 mutations have been found in squamous cell carcinoma. Those carcinomas are caused by the keratinocytes are involved. And by using this model, we are really very interested in studying how different variants can probably affect signaling in this particular cell type. Apart from the hematocylin eosin stain, the hematocylin eosin stain basically provides you with the dark blue is the nucleus and the light blue or pinkish is the cytoplasm of the cells. But we can also do immunohistochemistry with this epidermis, and then study particular proteins that are involved in the cell cycle division. So you can measure whether or not the cell is triggering mitosis, and there are also markers for differentiation and so on. So you name it. There are a bunch array of immunoantibodies that can be used to study these membranes. And last, I would like to end the presentation by presenting the idea in the midterm, although we already started, but it's very preliminary. The collaborations. One, the top one is with the Iowa State University in the United States, where we're going to use zebrafish model to test in vivo our DNA variants. And then here in France, in Paris in particular, in the Institut Jacques Monod with Vanessa Ribes, we are going to use the chicks embryos as the model, as an in vivo model to also study our variants. Mario is one of my PhD students, and he's here in Paris with me. He will stay for a month in Vanessa Lab, so also feel free to go to talk to him if you are interested in this field and this topic. These two models are very good to measure RAS map readouts, such as phosphorylation in several biomarkers of the pathway, expression of certain genes, and also measure proliferation of cells, embryonic development, and migration of certain type of cells. That being said, the key takeaways of my talk would be that basically my lab do basic research in which we can study DNA variants that have been identified in pediatric patients with RASopathies. And the good thing is that after these 2 years where we had to start from scratch as a new lab, we've been able to see that the reasoning behind all our ideas is working, that we can definitely generate these stable cell lines, and that we can measure all these different biomarkers to study further and deeper what's the molecular mechanism behind those variants, and try to identify genotype-phenotype correlations in the variants. And the overall idea will be, hopefully within a year or two, to have all the data and then put together the in vitro, the in vivo data, also the analysis with the different drugs, and try to find a more personalized precision medicine for these patients. This is a picture of the cathedral of the city of Salamanca. So I really invite you to Salamanca. You are very welcome there. Thank you so much for listening. I hope it was of your interest, and I'll be happy to answer questions, or I'm open to discussion also with you. So thank you very much. Merci beaucoup.
Thank you very much, Jesús, for your talk. So I have already a question from Vanessa Ribes. So she says, hey, Jesús, how come variants in a single gene associated to the same type of disease have distinct effects on MAPK activation, per K, in parentheses? Other pathways affected? Is there more heterogeneity than expected in RASopathies?
So the first question is basically because depending on the location of the variant, different domains of the protein will be affected. And then depending on the protein, that variant can trigger different effects on signaling. As an example, if you think of neurofibromin, it's a really large protein. It contains at least 6 known domains, and each domain has its own interacting partners. So therefore, depending on the type of variant, it will affect more or less downstream proteins, or even possibly upstream proteins that regulate the protein, and the effect will be different. So that's, for now, the rational explanation to answer that questions. And then the second question was more regarding the heterogeneity.
Yeah, is there Is there more heterogeneity than expected in the rhizopathies?
It is, and it's because I think we're still far from understanding how the RAS MAP signaling pathway is regulated. There are many other pathways involved in which RAS is involved, and that's probably why, because we still do not know other feedbacks and other loops that are involved in these pathways. And despite it seems pretty straightforward signaling pathway from SHP2 to ERK or to mTOR, there are still many other players, and we do not know fully. We do not fully understand how all those other players are interacting with the proteins that are part of this signaling pathway.
Thank you. So we have another question from Vanessa. She says, what about the impact of cell types on the response of cells to variants associated to rhizopathies?
Well, you can have from migration to more migration, less migration. You can have more proliferation. You can have less proliferation than the controls. You can also have the size of the cells is different. If they trigger mostly when they affect ERK, what you have is what they call a transforming morphology. That means the cells change their shape. They are like more elongated, and also the way the light goes through them is different. They are kind of translucent. So there are different physical or morphological parameters that you can quantitate to compare the effect of the variants on the cells.
We have another question from Boone. So he says, thank you for your excellent talk. Do you have patients with typical manifestation for syndromes, but no mutation candidate genes were found? If so, how do you approach these patients in terms of diagnosis?
Well, so far, in total, we have 110 patients, and for all of them, from the hospital, they've been able to identify the genetic cause, like in all cases. But that being said, in some cases, the identified mutations are, let's say, uncertain, because either they are completely new and therefore there is no information regarding whether it's just a low prevalence or a low abundant human variant that could be possible without being pathogenic, or whether it is a real pathogenic variant. So that's one thing. In the event, as the question was targeting, if you don't find any change in the DNA, then you would apply different cytogenetic techniques to see whether you had rearrangements in chromosomes, or if you had a gain or loss of some part of chromosomes, basically, which could lead to identify possible genetic information missing in those cells.
So we have a question from Pascal Giraldi, which says, in neurofibromatosis patients, one allele of the NF1 is mutated, and generally the second allele is deleted in the tumor. Can you reproduce this second event in your cell culture?
We could. So Pascal is right. Usually patients who only have one allele mutated, they develop neurofibromatosis type 1. And normally, in order for the tumor to develop, they need a second allele mutated. And we could reproduce that. We could, starting by applying CRISPR and maybe deleting one of the alleles of the gene, and then either generating another, using CRISPR to change the other allele, or using that background to transduce the cells with the variant that you want to overexpress. That could, these two approaches could be probably the best one to do that. In any way, you would have to consider that whenever you are using viruses to express a gene, you are overexpressing that gene. And that also you always need to take into account, because the overexpression itself, it could have an impact. Okay, and in patients, you don't usually have overexpression. You have a mutation, and then that mutation is enough. And in our case, we're overexpressing. So that's also that we always need to take into account. Okay.
So we have a question also from PR. So he said, "Thank you for your talk. I truly enjoyed it, and it was I was inspired by how your work can impact patients' lives. I would like to ask a personal question. When, if in your career, did you have a wow moment when you were surprised or impressed with your research? Did you have a moment that changed you forever and led you to where you are today?"
Can you repeat that again, the question?
Yeah, sorry. So he says, if in your career, did you have a wow moment when you were surprised? Yeah, if you were surprised or impressed from your research, and did you have a moment that changed you and led you to where you are and what you are studying right now?
Well, I do have one. It did not necessarily drove me here, but I really have one, and since there are probably a lot of students here, and I was a student until just a few years ago, I think, and I recommend to always be curious. Be curious about what you're doing in the lab. Always pay a lot of attention at what you are doing and at the results you are having, because one of the stories I can tell is that I was working with this bacteria, actually in the United States, in Davis, where I went for 3 months. And one of, depending on the week, I would say, the experiments were one result or another. I had like 2 different results, depending on the week. And at that moment, we had no clue what was going on. And my supervisor was kind of blaming me of maybe doing things differently. And I was like, no, I really promise I'm doing everything in the same way. I don't think it's me. But despite that, it's like my supervisors, they lost the interest on the results because they were not consistent. But I continue working on that. And eventually, to make a long story short, what was happening is that some bacteria, over time, after a week, they were losing a megaplasmid that they were carrying, and that megaplasmid was containing genes that were essential for the chemotaxis assays that I was doing. And thanks that I was very persistent and that I was, believing in what I was doing was right, and I was super curious about that phenotype that was changing in the cells, we published actually many papers with that study. We identified the megaplasmid. We identified the chemoreceptors responsible for chemotaxis in those bacteria. And I felt like, wow, I'm happy that, I did not listen to my supervisors, and I continue doing what I felt it was the right thing to do. So that's kind of the story.
Yeah, a great story. Okay, so I think I have a last question. It's from Mohamed Kaili. Says, "Very interesting talk, thank you. Did you find variants located in non-coding regions? If the genes of interest involved in RASopathies, and if yes, how these variants affect the RAS pathway?"
Yes, absolutely. The most abundant non-coding mutations that we find in the RASopathies are those that affect the positions that are conserved in the introns. I do not remember. I don't know if you remember that the introns contains the first 2 positions on the 5' end, and the 2 last positions in the 3' end are conserved. And if a mutation affect either one of these 4 positions, then the spliceosome cannot take that intron out, and that is going to affect the synthesis of the protein. And sometimes it even affects the expression of the translation of the mRNA somehow. We still don't know how. So absolutely, yes. And despite on top of mutations in those conserved introns, in those regions in the introns, there are also mutations in like promoters that can affect the expression of these genes. And there are also less known, but there are also mutations in introns that somehow affect at the transcriptome level, how much mRNA or how many mRNA the cells are producing.
Okay. So I think it's all the questions we have, and we have passed the time, so it's perfect. So thank you, Jesús Lacal, for your talk again.
You're very welcome.
Thank you everyone for coming. Please do not hesitate to contact Dr. Lacal. We have sent his email. So if you want to contact him, you can. We will have the next edition of the seminar series on April 4th. So we are not sure for now who will be the talker, but we will contact you when we know, and we hope that you will come also next time. So I don't know if Madeleine wants to add something. If not, then thank you everyone for coming, and I will let you leave the conversation. Thank you.
Yes, thank you very much. Have a great evening, and hopefully see you all next month.
All right.
Bye.
Thank you very much. Bye.
Bye. Thank you.
Take care.
Goodbye.